We're going to take another sort of little turn. Again, we've gone through some stimulation and then some regeneration and regrowth, and now we're going to go into a territory that I'm guessing a lot of you haven't heard about or thought about relative to the effects of spinal cord injury. And I was just thinking about it just in the last minute. My son's had an injury for 11 years, and like Jason yesterday talking about UTIs and the frequency of them, in the early years he had that frequency. He hasn't had a UTI in years, not because he changed anything with his bladder care, but because he eats a pretty whole food diet and doesn't drink any soda. He does drink some other things occasionally, but he's very attentive to his gut. And as we are, I think, learning, our guts are responsible for things that we didn't previously understand. And so relative to that, these next two presentations are going to be in that area. So please welcome Dr. Phil Popovich.
How about now? Perfect. Thank you, Matthew, for the introduction and the invitation to present today. As Matthew indicated or alluded to, the next two talks are going to go into some areas that perhaps you've read about, but my guess is that if you've read about it, you've not read too much, because there's not a lot of literature on these next two topics, especially in the context of traumatic spinal cord injuries. So we hope to stimulate you and get you to be thinking about some things that may be affecting you directly that you don't even realize, that the microbiome and the liver and liver dysfunction are at play and contributing to some of the things that you're dealing with on a regular basis. So we spent a lot of time today or this morning and yesterday talking about the effects of spinal cord injury on locomotor dysfunction, sensory disorders, or sensory disturbances, pain in particular. What we haven't spent as much time doing is talking about many of the other comorbidities and problems that affect individuals with spinal cord injury and their families as well. So this concept that spinal cord injury is more than paralysis is not something that's new to you, but it's not something that the research community as a whole has spent a lot of time thinking about. And I believe, and I believe Dana believes in the next presentation, that a lot of these other comorbidities that affect spinal cord injured individuals are affected by changes in the gut microbiome, and as well as the ability of the gut and the gut microbiome to communicate with the liver and then with the rest of the body systems. So at Ohio State, we have a large group of individuals now who are trying to study and understand traumatic spinal cord injuries from a systems-wide perspective, meaning that the process of traumatic spinal cord injury sets in motion a slow disease process that affects organ systems throughout the body, and that can have implications for not only locomotor and sensory recovery, but also how the rest of the body responds to the injury and how it ages, how it deals with many other things that happen on a regular basis, the drugs that you take, the ability to metabolize those drugs, the ability to extract nutrition in the appropriate way to fuel your cells throughout your body so that your organ systems properly function. So these are the types of things that I want you to be thinking about, and I'll bring them back throughout my talk today. So the first two questions that we're going to ask and provide some answers to in the beginning of this talk is, what is the microbiome, and how can microbiome research help me? So let's start with the first question, what is the microbiome? And to do that, I actually need to introduce the concept of microbiota. So the microbiota is the community of microorganisms that live in us and on us. So in your skin, all of your epithelial, or all of your mucosal linings, your mouth, primarily your gastrointestinal system, are lined by a series of bacteria, viruses, fungi, archaea, and protists. Okay, so we are carriers for microorganisms. In fact, it's been estimated that more than 99% of the genetic material that lives in us and on us is derived from microorganisms. So if you think about that, flip it. Less than 1% of the genetic machinery that exists within you is mammalian. Okay, so we are huge carriers for bugs. Now, the microbiome is the genetic component of the microbiota. So oftentimes you'll see those words used interchangeably in the literature. And so I just want you to understand the subtleties between the two of them. So why should I care about microbiome research? How can it help me? And for us to answer that question, you really have to understand what these gut microbes can do. Okay, and I'm going to focus exclusively on gut microbes. And again, that's because most of the microbiota live within our intestines. So it's helpful, I think, to think about the gut microbiota as this important integrator of all the information that comes to the body from the outside. And that can come in the way of stressors, so intense, prolonged periods of heat or cold is a good example, or the food, the thing that's probably has the greatest day-to-day effect on the microbiota are the foods you eat. All right, so as these signals come into the body, these microorganisms that live within the gut, they interact with those signals by a variety of different ways, and then they interpret those signals, they metabolize and degrade those signals, and release a variety of metabolites and different types of molecules that go into the bloodstream and influence cells throughout the body. Okay, and all this happens without you really thinking about it. The only time that you realize that it's happening, or the importance of this microbiota, is when things change in the gut, or things change in your diet, or things change in your external environment. Everybody in this room has experienced a medical condition, whether it's an acute or chronic condition, that's been related to a change in your gastrointestinal system. And that is by virtue, largely, of the changes in the microbiota. So why is it important to study the microbiota in the context of spinal cord injury? Now, this is an important question because years ago, when the microbiota research field started to evolve, okay, and it's been around for a long time, but it's only in recent years that it's become a very acute of acute interest to the medical community and research community. This is a schematic from a paper that was published, I think, about 4 years ago now. And what it shows is that disruption of the microbiome has now been linked to diseases that affect the brain and all organ systems throughout the body. Today, we now know that virtually every disease that you can think of has been linked back to a change in the gut microbiome. Now, cause-effect relationships are things that are very difficult to prove when it comes to changes in the microbiome and how it affects body systems. I'm happy to talk about that later. I'm not going to be able to go into detail about it today, why that's so complicated. But proving cause-effect is difficult, not impossible, but difficult. Given the proved importance of the microbiome, or the proved association between changes in the microbiome and the rest of the body, we became interested in asking whether or not spinal cord injury will cause a change in the microbiota. And does it cause a process called gut dysbiosis? And gut dysbiosis is simply a change in the balance. Okay? You have these microorganisms, these bacteria, viruses, et cetera, living in your gut. And under normal circumstances, they live in harmony, okay, in a process called homeostasis. When changes happen, chronic changes happen to the gut microbiome, they go into imbalance. And that imbalance is characterized by an increase in bacteria and viruses, specifically, that can cause problems, pathology, and a decrease in the bacteria that actually are beneficial to the physiology of your organ systems throughout the body. So a few years ago, we published this paper. This is a preclinical model of traumatic spinal cord injury in mice, in which we proved definitively that gut dysbiosis does occur after a traumatic spinal cord injury. And we used a model of mid-thoracic spinal cord injury. It was an incomplete lesion, a severe incomplete thoracic spinal cord injury. And we were able to prove, using a variety of techniques, that gut dysbiosis develops. It takes about 2 weeks for it to develop and become prominent, and it never goes away. Now, in this paper, we showed a number of things that I'm not going to have time to go into details on, but we showed that the dysbiosis actually has a negative effect on spontaneous recovery of locomotor function. So it affects the locomotor system in ways we don't understand, but it affects them. Okay? It activates the gut immune system. Why is that important? Well, if your gut immune system doesn't work properly, a lot of the foods you eat will introduce pathogens, which will cause you a great deal of gastrointestinal distress, hospitalization, and under worst-case scenarios, even death. So it's important because our gut immune system, called our gastrointestinal associated lymphoid tissue, or GALT for short, It's critical for helping protect us from an external environment and also communicating to the rest of the immune cells throughout your body. So your overall health is dictated by the ability of the microbiota to control immune function in the gut. We know that a dysbiotic response after spinal cord injury actually exacerbates the neuroinflammatory reaction that develops within the spinal cord, and you heard a little bit about that today from Lana's introduction to the different cell types that are affected after a traumatic spinal cord injury. So just tweaking and changing the microorganism in the gut amplifies the neuroinflammatory reaction and actually makes pathology caused by trauma worse. And then at the end, I'll talk to you a little bit about, the good news is that we think it's possible to try to restore some normality to this gut dysbiotic response. And I'll talk a little bit about our work with medical grade probiotics. Now, this was all in a preclinical model, a mouse model of traumatic spinal cord injury. However, it's relevant to humans as well. We now know there's a number of different studies that have been published over the years showing that gut dysbiosis occurs in people, and it occurs chronically. There's been examples where samples have been collected from spinal cord injured patients many years after their traumatic spinal cord injuries, and there's still evidence of profound gut dysbiosis. What we really don't understand still is what is the functional consequence of that dysbiotic response, and that's where I'm going to try to get into a little bit as the talk moves on. So many of you may be asking, okay, that's interesting. Gut dysbiosis occurs. You can prove it in a preclinical model. Why does it happen? So I talked a little bit about how environmental stressors and food can contribute to this. There's a lot of things that actually can trigger gut dysbiosis, and many of those things affect people with spinal cord injury on a regular basis. But we believe that there's, at the heart of a spinal cord injury, a very fundamental reason why gut dysbiosis develops and why it persists indefinitely. And that's related to how the autonomic nervous system innervates and controls gastrointestinal function. So this schematic here illustrates the brain, brainstem, spinal cord, and then the connections that emanate from the brainstem and spinal cord predominantly to the gastrointestinal tract. So these are different linings of the gastrointestinal tract. This would be the lumen or the center of the gut, and these would be the muscle walls of the gut. And you can see all these different colored lines that are coming off of the spinal cord connections. They directly are innervating and communicating with the gut, both the muscle wall and the mucosal layer. Now, after a spinal cord injury, what happens, if the damage occurs in here, you can see that the connections that are emanating from the cord and the brainstem are lost or damaged. And when that happens, it creates a condition called neurogenic bowel. And I'm sure many of you in this room, if not all of you, have some disturbances with your bowel. This imbalance due to imbalance in autonomic control, there's an imbalance in the tone between what's called the sympathetic and the parasympathetic nervous system. And when these two parts of your autonomic nervous system go into imbalance, it changes the tone to the gut, and it changes motility, it changes mucus production, it changes the ability to absorb nutrients from the gut lumen from food we eat. Now, in the context of spinal cord injury, it's also important to understand that this autonomic tone is regulated on a segmental level. So that if the spinal cord injury occurs up here in the cervical or upper thoracic level, the ability of the brain and the brainstem to communicate with the autonomic neurons that live down here to the gut are all disrupted. Okay? That entire circuit, that feedback circuit's gone. But if the injury is down here a little bit lower, let's say mid-thoracic or even lumbar or sacral spine, a lot of this autonomic tone can persist and may still be abnormal, but it's more controlled than if the injury had occurred at a higher level. So one of the outstanding questions in our preclinical models, also in the human literature, is whether or not there's a level-dependent effect on the gut microbiome. Does it matter where the injury occurs to cause this gut dysbiosis? So we began to start studying this again using our preclinical models, and this is really one of only 2 data slides I'm going to show you in this presentation. And what you're looking at here are data that were developed by analyzing fecal samples using a technique called whole genome sequencing or shotgun sequencing. And what this technique allows us to do is identify the DNA, all DNA, mammalian and microbial DNA in the fecal samples, and then using computerized databases and algorithms, we can tell the nature of all the microorganisms that exist within these samples. Each of these colors in each of these bars, so each of these columns represents a single animal from our control group, our mid-thoracic injury group in green, or the high thoracic injury here. Okay, so there's 5 animals per group, and each of these colored bars represents a type of bacterial species. The larger the bar, the more there are of that species. So you can see in the sham-injured animals, there's a lot of this purple bacteria, which is called a lactobacillus. You can see that after a spinal cord injury, there's very little of that bacteria left. But you can see that in the high injured group, high thoracic injury group, there's more of this orange bacteria called a Weissella. So this is, again, evidence of gut dysbiosis, but it's also evidence that injury level impacts the composition of bacteria. So presumably, the functional consequences of the gut dysbiosis will change as a function of injury level. Okay, so that is why we think spinal cord injury causes gut dysbiosis, again, related to autonomic tone and imbalance in the onset of a neurogenic bowel. What are the consequences of gut dysbiosis? Here's where we're going to do some hand waving, and I'm going to show you some examples of how dysbiosis can contribute to functional impairment in different contexts in the body. Now, again, because we're using whole genome sequencing, and we have all the DNA material and all the genetic code that's occurring within these samples, we can begin to use the same databases to predict function associated with bacteria, because we know everything that they're able to encode through DNA transcription. When we use a technique, in this case, what we're doing is we're creating a heat map where we're making estimates based on the DNA code of the bacteria that persist. We can begin to evaluate different types of functions, which are shown here along the left. And what this is called a heat map. The reds and the yellows are warm colors, representing there's high function associated with these different bacteria in each of these rows. And these functions you can see in the sham-injured animal, most of these functions are warm, meaning that they're active. But look what happens after mid and high thoracic spinal cord injury. A lot of these functions go away. Now, what do these functions mean? What can they mean at the cellular level to influence your organ systems? Let's just focus for a minute on amino acid biosynthesis. Tryptophan metabolism and the breakdown of tryptophan from foods that we eat is incredibly important for providing the precursors that we need for a variety of different functions. So here's a pictorial to really illustrate that. This is the gut lumen. This is where the food is coming through, right here. And you can see these microorganisms, one of them called a lactobacillus, which I told you is disappearing after traumatic spinal cord injuries. As the food is metabolized and tryptophan is broken down by the lactobacillus, it produces a variety of metabolites that are necessary for basic physiological function. This next slide really just shows you an illustration of the many different things in the body that require tryptophan metabolites. This is the gut lumen. You can see here the bacteria. This is tryptophan being metabolized by the bacteria. And this is just showing you the variety of different metabolites that are used by different organ systems in the body: the brain, the immune system, the liver, as you'll hear more about in a minute, kidneys, the gut immune system. These tryptophan metabolites are needed everywhere. And after a spinal cord injury, these lactobacilli are gone. So tryptophan metabolism is significantly impaired. It's starving cells. The absence of these bacteria starves cells throughout the body from having this essential metabolite. Lactobacilli and other bacterial taxa also produce other metabolites called short-chain fatty acids. You may have heard of butyrate or butyric acid. When these are lost or diminished, it has wide-ranging effects in the body, including a change in how the immune system functions, the immune cells throughout the body, your ability to fight off infections. Viruses, bacterial infections are impaired in the absence of these short-chain fatty acids. Insulin sensitivity, which you'll hear a lot more about in the next talk. And the ability to store energy and have cell anabolism, cell buildup, cell hypertrophy. All those things are impaired in the absence of these short-chain fatty acids. Why is that important? Well, here's a context in which it could be very important that we never talk about. So the concept that dysbiosis can impair or contribute to pathology in the gut-muscle axis. We all know that people who have a spinal cord injury and other types of paralysis lose muscle mass. And we often talk about that's due to primarily unloading of the muscles, no longer being able to prevent force and cause contraction, the hypertrophy of the muscles. But the reality is that there's also muscle mass in other areas of the body that aren't affected by the loading. we know that short-chain fatty acids are important for the muscle cells to undergo an anabolic state. So again, a starvation hypothesis is at play here as a result of gut dysbiosis. The good news is that despite the fact that these changes are long-lasting, I think there are ways that we can potentially begin to intervene and manipulate the dysbiotic environment. And one way in which we've started to do that is through the use of probiotics. Now, I'm not going to go into the rest of this, but there are other ways to potentially manipulate the microbiome over time. What we've tried in the laboratory is using a medical-grade probiotic. At the time we did these experiments, this probiotic was available commercially. I think it still is, although it's not the same formula. It's a probiotic called VSL number 3. And what's interesting about this probiotic is it contains a large composition of lactobacilli. So when we treated spinal injured mice beginning post-injury with daily doses of this VSL3 probiotic, we were actually able to improve locomotor recovery in these animals, which was something that blew my mind, and I didn't believe it for the first time that we did the experiment. We actually have done this experiment 3 separate times now, and we get the same results every time. It's not a huge increase in locomotor function, but it's a consistent increase. It tells me that something that the lactobacilli and or other components of the probiotic administration are fueling something that's important for locomotor function in these animals. It could be the muscles. It could be some precursor that's necessary for coordinating normal neural function in the spinal cord. Tryptophan is an important metabolite for serotonin biosynthesis, for example. The reality is we don't know why it works. All I can tell you right now is in our preclinical models, it works consistently. Now, I'm going to end with this last slide because this really introduces Dana's talk, but it also shows you the integration between body systems, organ systems that are affected by spinal cord injury. Over here on the right, this is the gut lumen. This is where the microbiota are. After an injury, when the dysbiosis occurs and there's a change in the metabolites, the ability of those metabolites to coordinate communication with the cells in the gut lumen that keep the bacteria in the gut, they change and they actually become leaky. So the gut epithelium can leak. And when that happens, these bacteria can actually leave the gut and enter into the portal circulation. So this concept of bacterial translocation has been proved in animals and in humans. And when the bacteria leave and get into the blood supply, they drain directly into the liver. And there, they can trigger inflammatory reactions, which again, Dana will talk to you more about, setting up this reality that changes in the gut trigger pathology in other regions in the body. And all of this is accelerated by traumatic spinal cord injuries. So with that, I'm going to leave you with that thought and to set Dana up, and I want to just briefly acknowledge the members of our large spinal cord injury center. I want to introduce Matt Sullivan. Matt Sullivan's my collaborator on the metagenomic work that I briefly talked about. And Chrissy Kiggerell, who would be over here on this side. She's been in my laboratory for a long time, and Chrissy's led most of our high-impact research projects, and she's been the driving force behind all of our microbiota research since its inception. And of course, many of our funding organizations that have supported our work, in particular the NIH, has supported all of our microbiome research to date. Thank you very much for your attention.
Thanks, and now welcome Dr. Dana McTigue. Teague? Tige?
McTigue, but it looks like McTigue.
McTigue. Okay.
I answered anything. Alrighty. Hopefully you can see me. I'm much shorter than Phil was, so kind of have to stand on my tippy toes. But so thanks for staying and coming to our session. Like Phil said, this is a topic that you probably don't hear much about and maybe probably don't think much about. I know I didn't start thinking about liver pathology when I first started doing spinal cord injury research almost 30 years ago, I guess it was now. This is something that we kind of came upon by accident, just serendipitously in my laboratory, and it kind of really melds well with what Phil's been working on too. So I'm going to talk to you a little bit about how spinal cord injury affects the liver and what this does to the rest of your body and why you maybe should care about that. So since we don't typically think about our liver very much, I thought I would introduce one of the main things that the liver does And that is anytime you have a major trauma or a big injury or even a major infection, one of the liver's jobs is to try to get the body back to homeostasis. And the way that it does that is by doing something called the acute phase response. And in this response, the liver then starts to pump out into the body molecules like cytokines to fight off any potential infection. They send out molecules like fibrinogen and other molecules to form blood clots to stop bleeding. So the whole purpose of the acute phase response is to try to stem off the injury and get you back to normal and make things, make you safe from any potential pathogens. So what we notice, though, also happens whenever you have a spinal cord injury or whenever you have any type of injury, this acute phase response also causes liver inflammation. So as the liver is pumping out these molecules to mobilize immune cells to the injury site, these immune cells also come into the liver itself. So the liver undergoes some acute inflammation. But typically, that's not a big deal, and it resolves within a day or two. What we noticed in spinal cord injury, however, in our animal models, is that this acute inflammation is persisting chronically. So it's never resolved. So the liver is maintaining this inflammation in this high inflammatory state, which is going to impact its ability to do the other functions that it does. So our liver is really a pretty amazing organ. It regulates all of our lipogenesis and glucose and carbohydrate metabolism. It forms clotting factors. It does a lot of different things that we typically don't need to think about. But once the liver is maintained in this inflammatory state, it's not going to be able to function as well. So I'm going to show you some of the data, but not too many data slides, but some of the data to show you that we've discovered in our laboratory looking at how the liver changes over time after spinal cord injury. And so one thing is within the liver, there's a group of cells called Kupffer cells, and these are just macrophages. The liver actually has the most macrophages in the body. 80% of your macrophages are found within the liver. And that's because, like Phil said, all the food and particles from our GI tract drain right to the liver. So the liver is kind of our first line of defense. If anything comes out of the GI tract, these macrophages are there and they're ready to take care of it. So these macrophages are what become inflamed after spinal cord injury. And this is an example of 2 rat livers. This is from an uninjured animal, and this is a spinal cord injured animal from 6 weeks post-injury. And this is a stain for the Kupffer cells. So all these little black dots are the macrophages that are found within the liver. And you can see out to 6 weeks after injury in this rat liver, there's still many more macrophages, and they're much more hypertrophied, so they're much more activated. Whenever we quantify these changes in the laboratory, you can see in this graph, this is just the amount of Kupffer cell area, and injured animals are shown in black, and naive animals are in these white bars. And so we can look as early as 3 days post-injury, there's a massive increase in the Kupffer cell activation, and out to 6 months, this is still significantly increased. And we now have data in the lab out to 2 years post-injury in rats. And we see that these Kupffer cells never tend to calm back down, they always seem to stay activated. One of the things that the Kupffer cells do is produce cytokines and inflammatory molecules. So these are just some measures of different cytokines found in the livers after spinal cord injury in our rat model. And I know there's something with the projector. I know these guys are working on it. I don't know if you need to do anything. But you can see early again, within 3 days, we have a significant increase in these very pro-inflammatory molecules within the liver. And these are maintained out until at least 8 weeks post-injury. So these molecules, as you can see, these are pro-inflammatory molecules. So as these are up, they're going to contribute to the inflammatory state in the liver, and that's going to produce more inflammation, and so more pro-inflammatory cytokines. So this basically sets off a vicious cycle where the liver becomes inflamed, and then it keeps on keeping itself inflamed. Another interesting thing that happens in the liver is it actually accumulates fat. So our liver is capable of storing lipids normally. And it usually has a little low level of lipids in it. And so these are also some liver sections from rats after spinal cord injury or naive, and they're stained with something called oil red O. And this is just a stain that accumulates in lipids, so it accumulates in fat molecules. You can see normally you see a couple little blobs of fat in the liver because it's storing some low levels of lipids. But after spinal cord injury, this fat tends to increase. This is 21 days post-injury here, and we've seen this again out to 6 months post-injury. The livers are accumulating too much fat. And it's been shown, there's some data from spinal cord individuals that estimates of up to 50% of SCI individuals tend to have this fatty liver. This is a problem because elevated lipids can become lipotoxic, and it depends what kind of lipids are in there. There's some lipids that are very toxic to the liver cells, and so this constant overwhelmingness of the fatty accumulation and the inflammation can actually lead to liver damage. And so we looked in our animals for circulating liver function tests, so if we're looking for ALT. And so if you've ever had blood work done and they want to check your liver enzymes or your liver function, this is what they're measuring. This is an enzyme that hepatocytes make, And if a hepatocyte becomes damaged, some of this ALT just starts to leak out into the blood. So if you have ALT in the circulation, that suggests there's some low-level liver damage. And indeed, in our rats, we were able to see that that starts to happen about 3 weeks post-injury and is maintained for at least 8 weeks. This is relevant, too, to spinal cord injured individuals. A paper just came out, I think it's actually out now, where they were looking at markers in blood samples from spinal cord injured individuals at 3 months post-injury and 12 months post-injury. So they were looking early after injury, and then they were looking again later. And they were able to show that the best biomarkers for how the injury outcome was going to be was the levels of ALT in the individual's blood. So the more ALT there early, the worse the outcomes were at 3 months and 12 months post-injury. So this liver damage can be kind of a predictor of how well the recovery may go. This is just brand new data. So you may be wondering, okay, maybe there's fat in the liver, there's some inflammation. What does it really mean? What's the significance of these findings? This is actually a disease called NASH, or non-alcoholic steatohepatitis, which is just a fancy word for saying a fat liver that's inflamed that's not due to excess alcohol. Okay, so this NASH is what happens when people who have metabolic syndrome. So if you've heard of metabolic syndrome, this is whenever energy intake exceeds energy output. So basically whenever people eat too much and don't exercise enough, right? You tend to get increased obesity, and you end up with a lot of different problems with the metabolic syndrome. And so this NASH is what happens to the liver during metabolic disease. So this is happening just spontaneously after spinal cord injury. And NASH is important because NASH is part of a continuum from a normal liver to a fatty liver to liver with fat and inflammation. It can be a precursor to cirrhosis and to liver cancer. So how relevant is this liver pathology to spinal cord injury, you might be wondering? So one thing that we know, like Phil mentioned, the liver is the major organ for metabolizing drugs. And so we think that drug metabolism is probably altered in these livers that are pathological. So we've looked up, and there's just papers that are looking at different clinical trials using riluzole. So riluzole is a drug that's been tried in ALS patients and in spinal cord injured individuals. And one of the things you do in almost any clinical trial is measure liver function test. And so the papers reported that the patients in the ALS trial, and these, I should say, both got the exact same dose of riluzole. So they're both the same dose, and they're both a spinal cord problem, right? The ALS patients, only 4% of the patients had increased ALT levels, whereas 30% of the SCI ones did. So it suggests that this liver is maybe struggling a little bit to metabolize this drug. We also know, like I said, NASH is a precursor to cirrhosis. So hepatitis and cirrhosis are 7-fold higher in the SCI population compared to the able-bodied population. And these can also be precursors to liver cancer. And it turns out this is also significantly higher in the SCI population. And so these are the kind of things that kind of don't really get big news or not really flashy. They're not the same as recovering, motor function, but there's still significant problems and significant impacts on the quality of life in someone's health and someone's health span. So like I said, we know that this fatty liver is a manifestation of metabolic disease. So we wanted to go back into our animal model and see if they're exhibiting any other indices of having metabolic disease. So we went back to our animal model, to our time course, and we first looked in the blood samples for the levels of glucose and insulin. And we saw that both of these are significantly elevated over time in the animal models. And what this means is, just like patients who have diabetes, their glucose levels are too high. And what happens then is the pancreas starts spitting out more and more insulin to try to lower these glucose levels and get the glucose into the cells. But over time, the cells become resistant to insulin. They downregulate their insulin receptors, so the cells can no longer respond to insulin. So you have elevated glucose levels. And this is bad because excess glucose can be toxic. It can alter proteins in a pathological way. And also, our cells need glucose. So two of the major cell types that need glucose are the muscles and the liver. So the muscles are not able to take in their glucose. They're not able to function as well. They're not able to generate energy or generate ATP. So our animals are showing evidence of being insulin resistant, and this is the precursor to type 2 diabetes. We also saw in our animals another manifestation of metabolic disease, and that's excess serum free fatty acids, or this is just excess lipids in the blood. So whenever you go and get your cholesterol tested and your triglycerides tested, this is what this is measuring. And you can see that in our animals, again, over time it takes a while to build up, but they're also showing excess lipids in the blood. And this is a precursor to cardiovascular disease, as we all know, right? So once you get the fatty accumulations in your vessels, you'll get the inflammation in your vessels, and it can clog off and cause cardiovascular disease. And notably, some of these issues are much higher, again, in the STI population. So diabetes is 3 times higher compared to the general population. Myocardial infarction is 8.5 times higher, so elevated levels of heart attacks. Dyslipidemia, so excess lipids in the blood, is significantly higher. And these conditions, when they do happen in the STI population, tend to be more severe. They also occurred in earlier age. So STI individuals are suffering from these at a higher rate, earlier in the life, and they tend to be more severe. We also know that, interestingly, with some of these things, the injury location doesn't necessarily play a role with what types of metabolic disease happens. So no matter where the injury level was in a study with STI individuals, they saw similar adiposity and insulin resistance. And then actually injuries that were T5 or below tended to have worse lipidemia, so worse fat in the blood. We also know that STI is occurring at people at a later and later age, right? So people that might have come in with these pre-existing conditions already are getting spinal cord injuries, so that's going to very likely impact their outcome. And we also know that there's a paper that documented that 25% of people that get spinal cord injuries are obese at the time, so they already have metabolic syndrome. And another 25% are overweight. So they're already coming into this injury with less than ideal health conditions. So one thing I wanted to do recently in my lab is try to get a gauge of, okay, we see these metabolic problems, how bad are they really compared to obesity? So compared to something that we know causes major metabolic problems, well, we needed a frame of reference. So what we did was we did a study recently where we took some rats and we put half of them on a high-fat diet for 8 weeks and half of them on a control diet. Then we gave half of them spinal cord injury, and then we kept them on their diets for another 8 weeks. So we had animals that came into the injury already with metabolic syndrome, and they maintained their high-fat diet afterwards. And we wanted to find out what happens to their metabolic indices. We looked at a lot of different things in these animals, and some of the data is still being analyzed. We know their locomotor recovery was worse, but I was going to just show you the metabolic indices. So we saw the liver inflammation was actually just as bad. So in these graphs, we have our uninjured animals on a normal diet. And then these in the yellow are uninjured animals on the high-fat diet. And then these are injured animals on a low-fat diet. So we're just comparing the animals that are eating a high-fat diet to animals that got spinal cord injury. And you can see in every indication, the spinal cord injury was just as bad as the animals being on a high-fat diet as far as liver inflammation goes. So the cytokines and the activation of the macrophages were comparable. The same thing with the circulating factors, so elevated glucose and insulin. SCI caused the same amount of pathology as being on a high-fat diet. The same with serum triglycerides and fatty acids. So and actually, the triglycerides were worse in the SCI rats compared to rats eating a high-fat diet. So you can see that spinal cord injury is comparable, basically, to eating a high-fat diet in terms of what it's doing to metabolism. And as you might imagine, the animals that got the high-fat diet plus spinal cord injury, almost every outcome was worse. So I'm not going to show you all that data. But the combination of these two is even more pathological. So we know that there's metabolic problems happening after injury, and that it's just as bad. So we hear a lot in the news about obesity, and we need to, there's a lot of obesity in America and other countries, and we need to, get people off that because it's so dangerous for their metabolic health and the cardiovascular health and long-term health. But it seems like the spinal cord population, even without having this obesity, they're still suffering these same kinds of problems. So we're trying to raise awareness of this. And like Phil said, why does this happen? So one reason that this can happen after spinal cord injury is like these bugs that are living in your GI tract, right? The GI tract is drained by these veins, and these veins all coalesce into this one vein called the portal vein, and it goes directly to the liver. So every bug that comes out of the GI tract, and after injury, we know we're going to have more pathological bugs coming out, more dangerous bugs, more toxic bugs. They go right to the liver, where that huge population of Kupffer cells are sitting, right? Those macrophages. And so these bugs directly activate the macrophages and get them fired up and get them pumping out cytokines and pro-inflammatory molecules. So having these bugs coming out of the GI tract, it seems to be going on as long as we've looked. So that can be one of the reasons why we're having this constant inflammation in the liver. We also know the spinal cord injury, like Phil mentioned, can disrupt the autonomic input to the liver. So the liver is innervated by the sympathetic nervous system that's found in about the mid-thoracic level. And so whenever this input gets disrupted, the sympathetic nervous system, as you are aware of, like autonomic dysreflexia occurs whenever there's aberrant sympathetic outflow. Sympathetic nervous system gets activated for some wrong reason and dumps norepinephrine into the body. At the same time, it's going to be dumping that substance into the liver. And norepinephrine is actually quite pro-inflammatory in the liver. It causes the macrophages, again, and even the hepatocytes to secrete cytokines and molecules. So there's multiple reasons potentially for why this is going on in the spinal cord injured population. I'm actually submitting a grant Monday that's trying to investigate these mechanisms, so hopefully NIH finds it interesting and important. So just to kind of put it all together, what's happening after spinal cord injuries, we have the injury causes this liver pathology and this inflammation. We get systemic inflammation, which is going to be pathological to all the other downstream effects. We have also the injury causing indices of metabolic syndrome. This can lead to things like, one thing that happens actually, like Phil mentioned, is the muscle becomes weak. So in metabolic syndrome, not only does it become weak, when the muscle is insulin resistant, the muscle cells start to die off, and the muscle gets invaded by fat cells. I know these look like eyeballs, but they're actually supposed to be adipocytes, so these are little fat cells. And so the fat starts to accumulate within the muscles themselves. We also get these lipids put into the blood, and some of the lipids are very toxic, and some of these lipids called like ceramide can actually cross the blood-brain barrier, and they can cause pathology in the CNS. They can cause things like depression and degeneration and things like that. So low levels of ceramides in the blood are not very good for our CNS. We also know this causes the cerebral vascular disease and diabetes, and all of these can lead to increased mortality. And if you look on page 47 of your booklet, there's actually data in there that says mortality rates are declining due to things like cancer and bladder problems, but they're actually increasing for things such as endocrine, metabolic, and nutritional diseases. So this is why the SCI population has had a stable lifespan for the past 30 years. The lifespan is shorter than the able-bodied population, and it hasn't changed in 30 years. So we're getting good at taking care of some issues, but now other issues we're seeing are still cropping up. Okay, I feel like this all sounds very depressing, so I want to leave on kind of more of a positive note. So the good thing, I think, is that addressing some of these problems might be much more easy than trying to fix the spinal cord. Right? Our liver is right here. It's a huge organ. It's in the front of our body. These organs are going to be much more accessible, so they might be easier to manipulate. Once we know what these problems are, once we identify why the pathology is occurring, we might be able to come up with strategies then to try to get rid of some of these issues and make SCI individuals healthier, make their muscles ready, so whenever there's a device they want to get implanted, or there's a new therapy that comes along and you want to get out of that chair and get walking, we'll have your body ready and healthy and good to go for that. So I feel like these are additional targets for spinal cord injured patients and spinal cord injured individuals, and I think that we have an opportunity to intervene with those. So I think it's good we recognize this, but I think hopefully the future is going to be brighter. All right, so then I just want to thank my group as well. We have a great group at Ohio State University, and in my laboratory, Matt Goodis is my postdoc, and he's generated almost all of this data. So he's been an awesome postdoc, and I want to highlight the Nielsen Foundation. So we've gotten money from NINDS and DOD and a donation from the Belford family, but Nielsen Foundation really funded all this work because they funded my postdoc. My postdoc's been on a postdoctoral fellowship for a couple years. So Nielsen Foundation was able to fund all this work, so I really want to send out a big shout out to them. So thank you, and we'll be willing to take any questions.
I think maybe it goes without saying, but you should probably pay attention to what these two eat for lunch. Maybe. Of course you are. So let's take some questions. Lynn?
Okay, so 4 years ago I got C. diff. It was super not fun. Can my spinal cord injury have any impact on, so I took the antibiotic 4 times a day for twice a week, and 3 days after I finished, it came back. So then I did the tapering. Is there a reason that 5 months after I was diagnosed, it took a fecal matter transplant for me to get all better? Could my spinal cord injury have anything to do with that?
Hello? No? No. Nothing on that one. I already turned it off and unplugged it. So I'll yell.
No.
No? I can't yell? Okay, there we go. So C. diff is a whole different beast in terms of infection of the gut than the dysbiosis I talked about. That's a pretty significant pathogen. Fortunately, you did have a fecal transplant, and that's been shown to be very effective in managing C. diff. How your spinal cord injury affected it, I actually can't say. I don't even know. I think it's really interesting from a biological and research perspective to actually study that, but I don't have an answer for that. I can tell you, the idea of will antibiotics, and I know people have to take antibiotic courses periodically as a result of different types of infections that they develop, and will the antibiotics that will cause dysbiosis influence other functions? That to me is a really interesting question. It's something that I think we need to pay more attention to, because as I've been giving these talks, I've had people in the audience who've come up to me and said, when I was on antibiotics or when I was coming off my antibiotics, I had, all these other things happen to me, and they would describe them to me. And some of them were neurological. Some of them were linked, they think, to their spinal cord injury. But again, when they go in to get care, their physicians would say, well, we really don't understand that. We don't know why. And so I raised the question, is it related to the changes in the microbiota in relationship to the injury? I think that that's a wide open question that I don't have an answer to yet.
Hang on, he's coming.
Thank you. I've had 3 spinal cord injuries over the last 48 years. And so I've talked to a lot of different docs. And my gut doctor, what are they called? GI docs.
GI docs, yeah.
Yeah, so I've tried aloe vera juice, kefir, kombucha, all the various things, and he basically says you're wasting your money, that I should just take a really good probiotic. That's my first question.
So all the things that you mentioned have probiotic activity, and one of the things that, that slide is even misleading that I showed, because I showed a little yogurt container. There's definitely probiotic bacteria in yogurt, but you are not going to get the medical benefits of probiotics through eating yogurt. You have to eat so much yogurt to generate enough bacteria. And the thing is that those bacteria, once you ingest them, they compete with bacteria that already exist in the gut. And because after a spinal cord injury, you're dealing with a bacteria composition that's primarily a pathogenic, very rigorous and robust bacteria that does not want to be displaced, you actually have to outcompete those bacteria. So the medical grade high concentration probiotics are probably a better approach, but even that might benefit from having antibiotic depletion prior to taking the probiotics, so that you're basically recolonizing with a different composition of bacteria. Everything I'm saying to you right now, though, has never been tested, never been proved. So these are the kinds of things that in our preclinical models we want to address, because I think, as Dana alluded to, these are tractable problems that if I can come back and say, we've tried this intervention, and when we do this, it does make a difference in how those bacteria colonize. And now we're measuring function A, B, C, and I think it could benefit you in that context. But I can't answer that directly right now.
May I ask another question? All right. So most recently, I had a non-TB microbacterium, MAC, in my lungs that then traveled to my spine and started eating my spine. So my GI doc was really concerned that I had C. diff, and they keep testing me for that, but fortunately I don't. So maybe a question to you is, how much diet is proven, I guess, to really help fight this stuff? And yeah, does alcohol hurt? It's like the best painkiller on the planet.
Well, I can say that I know there hasn't been any studies done on diet intervention in spinal cord injury that I'm aware of, but there's a lot in the obesity field. So with metabolic syndrome, there's been a lot of studies looking at, can probiotics affect people with obesity and get their metabolic health better? And it seems like there's data to suggest it can. Again, like it's going to be these medical grade probiotics, and sometimes they're combined with something called a prebiotic. So you include the bugs, and the prebiotic is the food that the bugs like to thrive on, so you put them in together. And that seems to be able to lower some of the cardiovascular risk effects and some of the diabetes risk effects in the obese population. As far as just regular diet goes, I don't know, like there's general things like, the Mediterranean diet seems to be healthier, right? But I don't know that anybody's tested it rigorously, and I know for sure not in spinal cord injury. So. Oh, and the alcohol, I do drink wine every night, so that's all right.
All right.
I think one glass, one drink or two a day is okay.
So this is Anthony. Just on that same note, yeah, I do buy the Activia yogurt from Dannon in the Greek form. That's all protein and no fat, so can't go wrong there. I think that for anyone, I do work in the medical field too. I see CT scans of cancer patients all day long, and most of them are really obese. And with everything I've heard today, I think it's more critical, especially for our population, to really just watch your diet. Low-fat foods, don't overeat. If your liver's already in a rough state, don't feed it alcohol to try to filter out. And look at these probiotics. I mean, it's really important to us to stay fit and trim if we can, even by less calories if that's what it takes. But high fiber, high protein, guys, that's what I'm saying. Thanks.
Just really quickly, is there a medical grade probiotic that you recommend?
Actually, I actually can't recommend anything. I don't know. There really aren't a lot of medical grade probiotics that have been extensively studied. The VSL3 is the only one. If you go into the literature, you'll see there's probably more than a dozen clinical trials that have been done, mostly with infectious bowel disease or inflammatory bowel disease with VSL number 3, but there's a caveat to that, and that formulation has been tied up in legal battles with the original inventor of the formula, and it's no longer marketed. It's still marketed as VSL number 3, but it's not the same formula that was used in clinical trials or that I used in our experiments. So I'm not sure if it's available commercially now.
So I can answer this. I work with a group at the Cleveland VA that looks at pressure ulcers, and we're considering doing a study with VSL3. And our pharmacist identified this exact problem and did the research and found out that the original formulation is now being marketed by the inventor as Visbiome, Visbiome. So it does appear to be available.
Visbiome is the new name that was originally, and I knew that existed. I just wasn't sure if they were marketing it.
So I actually thought this was a really great talk, and the research that I do in pressure injury with Kath Bogie at the VA, we've recently identified that pressure injury risk of recurrence is higher in people who have fatty infiltration of muscles. So I think that when we think about the secondary complications of spinal cord injury and how it relates to diet, there's a lot more work to be done, but I think this might be an interesting link to explore.
Yeah, I agree. So there's also data to show that people with fatty liver disease are more prone to infections, just in general. So that definitely they can get more pressure ulcers and get more infections in general.
And then I had a question too. To what extent do you think that the amount of antibiotics that patients with spinal cord injury ingest impacts all of this vicious cycle and inflammation? Because we think of antibiotics as being something that's going to decrease inflammation by decreasing the infection, but are we in fact making things worse?
I think that the answer is I don't think we know. I can tell you my only experience with trying to address that question is in our preclinical models. We injured mice, let them reach a plateau in recovery, and then delivered a very robust antibiotic cocktail that had 4 different types of antibiotics in them. So we were really targeting most of the bacteria that we could with drugs. And when we did that, there was no gross change in locomotor function in these animals. But I can also tell you that I think that there's a critical window. And I'm not sure exactly what that window is, but I say this because when we treated with probiotics and we mapped changes in the bacteria in the gut, we saw that as we fed the probiotics, there was a spike in colonization of the gut at a time that was coinciding with an improvement in functional recovery. But then over the next couple of weeks, those bacteria that we're feeding, even though we're giving it to them every day, it's not colonizing the gut anymore. They're going away. But the functions maintain. If anything, they got better functionally. So it tells me there may be a window in which the metabolites that are produced by subsets of bacteria are positively influencing outcome. So I would guess the same thing is probably true if you wipe out bacteria during a critical period. It may have an adverse effect on function, but again, there's really no literature out there on it except our one paper.
And obviously, if you have an infection, you have to treat the infection, right? So it's kind of, you get a double-edged sword.
Question here. Obviously, a lot of this is tailored towards the liver and the metabolic process, so to speak, in the metabolic system. And you've referenced the eating whole foods. You mentioned that you're vegetarian. I'd assume that also the vegan option for a diet is also going to reduce inflammation. Talked about probiotics, but why is there nothing in the form of acknowledgement here of the reduction in the form of medications that people with spinal cord injuries take and the long-term effects on the liver, and namely incorporation of exercise to increase the metabolic process through metabolism, et cetera?
Yeah, so that's an excellent point. I actually have that in my grants, to convince NIH that this is really important. And there's other data to show that the spinal cord injury causes enzymes in the liver that are needed to metabolize drugs. Those enzymes are reduced, so you can't metabolize the drugs as well, which means the dose probably isn't correct. So someone with an impaired liver taking the same drug as somebody with a functional liver, the dose might be different. They might take the same dose, but the amount in their blood is going to be different. It might be higher in the pathological liver person. So that it is something that we're looking at in the laboratory. I think I've talked to clinicians, and they're like, wow, it's a really good point. We never quite thought about that. So I think that it's just, just now the awareness is coming. I mean, I've been working on this liver stuff for a couple years, and I swear 2 years ago, my colleagues who are interested in regeneration and all this, they'd be like, liver, who cares about the liver? We want to make axons grow. I'm like, yeah, well, you need a good liver to help, the body to be functional for everything else to work well. So I think that just the awareness is coming up now, and hopefully going forward, there's going to be more work looking at that.
One last question, John.
Hi, that was loud. First of all, 2 great presentations, and thank you both for exploring this very interesting area of spinal cord injury health. So Phil, question. You mentioned that the gut dysbiosis is a result of the damage to the autonomic system, the dysautonomia. And so as a result of that, you're getting non-optimal gut bacteria. And then, if I heard you correct, you said that the optimal way to repair that would be because there's such a strong, presence there, they don't want to leave. Take an antibiotic, get rid of the bad bacteria, repopulate it with a probiotic, and now you've got good bacteria. What I'm wondering, though, is in the long term, if you've got this dysautonomia that's causing this gut dysbiosis, wouldn't that good bacteria just turn sour and go bad eventually?
Yeah, that's an excellent question. The reality is we still don't know. So the chronicity of the gut dysbiosis and whether or not it's dynamic, in other words, the dysbiosis, once it starts, whether or not these compositional changes continue to fluctuate over time, hasn't really been studied. In the clinical samples and the clinical papers that have been presented, they're usually a single time point, and it's highly variable across the groups. They're clustered, but they're usually very highly variable. So we really don't have an idea about the dynamic nature in clinical populations. But we do, in our preclinical models, we do have some time course data that we're analyzing currently out to 6 months post-injury. And all I can tell you right now is I know is that there's dysbiosis persisting, but I don't know if it's dynamic. Now, back to your question about if we do use probiotics as an example to try to challenge and reconstitute a good bacterium into the gut. It's possible that you'll have to take the probiotics indefinitely for it to maintain a beneficial effect. However, that's only based on our one study. We don't know, and I alluded to this in answer to a previous question, what would happen is if I wanted to treat with probiotics, if I wipe out the dysbiotic environment and then started feeding the probiotics, would that recolonization hold indefinitely? I think that's an important question. We don't know the answer to that. Long term, I'm hopeful that by understanding the dynamics and the compositional changes in bacteria, we can move away from antibiotics and we can begin to use what's called a phage therapy. So these bacteria, they're maintained in homeostasis by viruses that also live in the gut. And if we can learn which type of bacteria dominate over time, even though they may be different in you and you and you, they may still be of the same phyla or family of bacteria, so that we can grow viruses that will specifically target that type of bacterial family, and it won't affect the other types of bacteria. So we can get to precision on targeting bacteria. We might be able to get rid of some of the side effects of systemic antibiotics.
Thank you both. This is complicated, isn't it? And particularly that question that you asked that triggered Dana's response, the system. Sometimes I think we get focused on the very narrow targets and forget that it's part of a larger system. And so I think it's a good reminder to us. And also it made me think a little bit about what I mentioned yesterday about relationships. That there's relationships within the science, the story of the science. There are relationships like, Dana, you mentioned. You want to make axons grow. I don't care about the liver. Well, maybe you should, because they're related. But so that relationship in the science, also, I want to remind you, the reason why we're here is because there's a relationship that you have with the science and a relationship that you have with one another. People who have a spinal cord injury, people who research it, people who want to make it a product, people who want to discover things, people who want to fund things that would help human beings. So just keep that in mind and keep remembering that we want to take one another seriously so that we can help to make sense of this system and make this system work for what we want. Barry's going to give you a couple of minutes to update on what he talked about yesterday.
So how are we doing so far? We all right? We're just about to go to lunch, and I thought I'd just remind you once again to please check out the booth space and the different tables that are out there. Many of our sponsors are representative, and this would not happen at all this weekend without the great support of our sponsorship. There's also a booth over there called NASCA, North American Spinal Research Organization. And you might see some reference to a thing called scitrials.org. This is a trial finder that's spanking brand new. So anybody in this room or know of anybody out there in the community that are interested in looking into research and spinal cord injury trials, not only here but around the world, it's a great resource, and we suggest people check it out. But really, we've heard a reference at different points throughout the weekend about a journey. And you look around the room here, and some of us are at very different parts of this journey. You break your neck or your back, you have a spinal cord injury. It is the beginning of a very long journey, and for some of us it's not been easy, and for some of us it's been a little bit easier. But the point being is, without these kind of resources like these weekends that we put together with Unite to Fight Paralysis, I think our journey would be harder. The more knowledge we receive, the better we're at navigating through this crazy world of ours. And it wouldn't be made possible without the sponsorship that we talked about. But the journey with Unite to Fight Paralysis doesn't begin and end on these weekends. It's going, like I said, throughout the full year. And there are many programs, many resources that are available on the website, and we urge people to look at that. But I'm reminding everybody that Unite to Fight Paralysis is not an organization of large means, and they have to fund these resources. And so to help us with our journey, we are asking you as individuals to look at helping Unite to Fight Paralysis with its journey in what's called Fuel the Journey. And Fuel the Journey is basically a monthly donor campaign. For those who want to figure out more about it, please check the website. But see Ryan at the back around the corner when you're registered. We urge people to somehow participate in it, because without doing that, we wouldn't be able to put on these great things and even get bigger and better. So again, enjoy lunch, and last sessions in the afternoon, but just to take up every moment you can get. Thank you.
Thanks, and just a couple more things. Thanks, Barry. One, to remind you, remember we have a survey. We want you to fill it out. It's on the app. please do it. I'm going to keep saying it. We really, really want to hear from as many of you as possible. Number 2, thank the volunteers who are here, doing a lot of little things like holding microphones and timing speakers and telling them you got 0 time left. And with registration, a lot of students from Case Western that Hunter mentioned yesterday, but also some sort of family friends of U2FP. So thank them. Also, you'll notice that some people have this red staff flag. Those are board members for U2FP and a couple of staff members. So also meet them, talk to them. Like Barry said, you can find out more about what we do. And also thank them for the work that has gone into putting this on. And of course, our sponsors. And I guess that's all I have written on my cup to remind me. So let's have lunch, and we'll see you back for some more.